In this module, we will discuss the DNA microarrays and microchips. And this is how scientists, as a matter of fact, the molecular biologists, they tackle the problem of dealing with a large number of DNA sample analysis. And in order to overcome this problem, the molecular biologists, they have adopted an inkjet printer technology to spot tiny volumes of DNA on a chip. Just as a inkjet, it sprinkles very, very small drops of ink to make a picture. That's the idea from where they get. So this allows many different DNAs to be spotted on one chip, which is called a DNA microarray. DNA microarray on a small thing. Now this system was developed by Vivian Xiong and his colleagues. And Vivian, they used a robot within 12 parallel pens, each of which can squirt out a tiny volume of DNA solution 0.25 to 1 nanoliter, which is equivalent to the billionth of a liter. So you can imagine that how small amount is required for this. Now these spots are very precisely and exquisitely small, only 100 to 150 nanometer in diameter, and the center of the spots are only 200 to 250 nanometer apart. So you can imagine the distance. And the end product, the results look like this, which is a schematic diagram of DNA microarray on a slide. The size you can see, it's almost 1 inch and 2 millimeter long. After spotting, the DNAs are air dried and covalently attached by ultraviolet radiation to a thin saline layer on top of the glass. So the DNA is fixed on the chip and just like a slide. Now another strategy adopted to reduce the size of a blot is by Stephen Fodder and his colleague. They pioneered the method in 1991 by using the same kind of photolithographic technique. They used the photolithographic technique where the computer chip was manufactured, and to build a short DNAs, that is the oligonucleotides on tiny closely spaced spots on a small glass microchip. That was the photolithographic. And in this diagram, you can see that how the growing oligonucleotides are on a glass substrate. In the first, there are some nucleotide, they are fixed, then masked, and they are subject to the light. So they are fixed. So again, there is a chemical coupling with that, and that's the first cycle. Similarly, in the second cycle, the same things are repeated with the light, and again, the nucleotide, it starts growing over there in the second cycle. And this cycle is repeated again and again to make longer nucleotide. Now, DNA microchip, that is the resulting chip, is known as a DNA microchip or oligonucleotide array. Nucleotide array because they are all arranged in a tiny slide in the tiny, tiny spots. Now, term DNA microarray can also be used for the DNA microchip. Now, this technology is very much miniaturized. Think about that about 300,000 oligonucleotides can be built on a chip only 1.28 by 1.28 centimeter, that is about half inch square. Imagine such a small area and where you can analyze about 300,000 oligonucleotide in one go. The process is so efficient that a set of a different oligonucleotides can be built in only 4 by n cycle. So you can multiply it by number of cycles by 4 and can build the oligonucleotide again on the slide. Now, oligonucleotide on microchips and cDNAs on microarray, they can be hybridized to the labeled RNA, isolated from cells to see which genes in the cell were being transcribed. So this is very important because in that microarray, you can see a large number of genes only in 1 inch square area and can get the result. Now the example is, for example, Patrick Brown and his colleagues. They used the DNA microarray technique to examine the effect of serum on RNAs which are made by human cells. And similarly, the microarray chip is used in agriculture, in medicine research, and in other areas. And it has given a very important and useful handle to the microbiologists to analyze a large number of DNAs and eventually a number of genes which are associated with them in one go. This technology is very, very useful, economical, and is used by large number of molecular biologists in the laboratories as well as by the biotechnologists in their laboratories to analyze the gene sequencing.